I want you to imagine, just for a second, that a tumor is a highly fortified castle.
Okay, a castle. I like that.
Right. And for decades, we've been trying to cure cancer by basically sending in the knights to bomb the battlefield. We find the castle, we look at the immediate moat and the walls around it, and we try to alter the chemistry right there on site.
Yeah, the local environment.
Exactly. But there's this groundbreaking 2026 paper published in the journal Cancer Cell by Liu and colleagues, and it suggests that we've been entirely focused on the wrong location this whole time.
We really have. It's a massive blind spot.
It is, because the tumor isn't just winning the war at the castle walls, it's actively hacking our immune system's supply lines while our defensive troops are still like miles away, down the highway in the bloodstream.
Which represents a fundamental shift in how we understand cancer biology. I mean, historically, we have had a highly detailed map of our adaptive immune cells.
Right, the T cells.
Exactly, the T cells, which, keeping with your analogy, act as the cavalry or the knights. But we've been completely in the dark about our innate immune cells. The infantry, essentially.
The ground troops.
Yeah, the foot soldiers. I'm talking about the myeloid cells, things like monocytes and macrophages, which actually make up the vast majority of the tumor microenvironment.
Oh, they outnumber the T cells.
Significantly. But we've known they're present, right? We can see them. But their origins, and particularly the timing of their programming, has been just a massive mystery.
Okay, let's unpack this for the listener. Why has it historically been so incredibly difficult to track these innate foot soldiers? Because when we track T cells, we can follow them easily, right?
Yeah, very easily. Because T cells go through a process called VDJ recombination.
Right, VDJ recombination.
So as T cells mature in the body, they physically shuffle their DNA. Think of it like shuffling a deck of cards to create a completely unique genetic barcode for every single cell. That makes them incredibly easy for scientists to track using standard sequencing. You just read the barcode.
But our innate immune cells, the macrophages and monocytes, they don't do that.
No, they don't. They lack that built-in nuclear genetic tracker. So it's almost impossible to tell which macrophage inside a tumor is related to which generic monocyte floating out in the blood.
Because they all look genetically identical in the nucleus.
Exactly. And what's fascinating here is that to solve this blind spot, the researchers had to stop looking at the cell's main nucleus entirely. They had to look elsewhere.
So wait, if the nucleus doesn't have a barcode, where did they look?
They turned their attention to the powerhouse of the cell, the mitochondria.
Oh, wow. Okay, going back to high school biology here.
Right. But understanding the origins and the family trees of these cells is crucial, because ultimately it dictates why some of our cutting-edge immunotherapies work miraculously for some patients and why they completely fail for others.
So how does the mitochondria actually provide a barcode if the nucleus doesn't?
Well, mitochondria possess their own separate tiny loop of DNA. It's completely distinct from the main human genome.
We call that mtDNA, right?
Yes, mtDNA. And this mitochondrial DNA mutates at a staggering rate compared to our nuclear DNA.
Why is it so fast?
Because it lacks the strict proofreading and repair mechanisms that protect our main genome. So random mutations or typos just happen frequently as the cells divide.
Okay, wait, I'm stuck on this. I want to push back here.
Sure, go ahead.
Because mitochondria are essentially like ancient bacteria living inside our cells. They are biologically super messy.
Very messy.
Right. So if their DNA is mutating constantly, and a single human cell can have hundreds or thousands of mitochondria, how on earth do you distinguish a meaningful lineage mutation from just random biological trash?
That is the million-dollar question.
It sounds like a computational nightmare.
It was, and it's exactly why previous teams couldn't map these cells. So to parse this data nightmare, the researchers built a customized computational algorithm called Mitotrek.
Mitotrek, like Star Trek, but for mitochondria.
Basically, yeah. And Mitotrek essentially prioritizes absolute biological certainty over broad sensitivity. It applies incredibly strict filtering rules to what we call the variant allele frequencies.
Which is just the rate at which these mutations appear, right?
Exactly. So first, the algorithm automatically discards any mutation found in more than 20% of the cells across a patient sample.
Oh, I see. Because, let me use an analogy here for the listeners. Think of it like a family recipe passed down through generations, right?
Yeah, the food analogies.
Right. So if a great-grandmother accidentally writes tablespoon instead of teaspoon for the salt, every single descendant who copies her version of the recipe will have the salty version.
Right.
So if a mutation is present in over 20% of the cells, it isn't a recent event from the tumor. It's a legacy mutation that probably happened when the patient was just a tiny embryo.
Precisely. It's too old to be useful. So Mitotrek filters out that ancient history to focus on the immediate timeline.
What's the second rule it uses?
Second, the algorithm strictly removes any cell that presents multiple conflicting barcode trajectories.
Conflicting how?
Well, in single-cell sequencing, sometimes two cells literally get stuck together in a tiny droplet by accident. It's a technical error called a multiplet.
So it looks like one cell with two different family trees.
Exactly. And Mitotrek ruthlessly trims those out. So by establishing these really rigid computational boundaries, they were able to use those random mitochondrial mutations as a highly accurate endogenous lineage marker.
Like an evolutionary breadcrumb trail mapping a cell's descendants.
Exactly.
But to read those breadcrumbs, the team didn't just sequence the mitochondria, right? Because they used a technique called mtDNA, which is a mouthful.
It is a massive acronym.
Let's break that acronym down for the listener because it's really the engine driving this entire discovery. So it stands for mitochondrial single-cell assay for transposase accessible chromatin using sequencing.
Right. And the key word there is ATAC.
I always like to use a mnemonic here. Think of ATAC as the attack of the hyperactive enzyme.
That's actually a great way to remember it.
Because basically, they use this hyperactive enzyme, a transposase, that physically jumps into the open areas of a cell's DNA and tags it.
Right. It can only tag the DNA that is unspooled and open.
So simultaneously, they are reading the mitochondrial typo to figure out who the cell is related to.
The family tree?
Yeah, the family tree. And they are mapping the physical 3D architecture of the cell's main DNA to see what instructions are actively being read.
It's dual-layered mapping, and they applied this to a staggering 218,715 individual cells.
That is a massive data set.
Huge.
Yeah.
And they specifically chose patients with early-stage non-small cell lung cancer, which tends to be a highly inflamed, mutation-heavy battlefield.
A hot tumor.
Right. And they compared that to ovarian cancer, which typically has a cold, immunosuppressive environment.
And where did they pull the cells from? Just the tumor?
No, and this is crucial. For each patient, they sampled 3 distinct zones. The tumor itself, the non-involved normal tissue surrounding it, which they call NILT.
NILT.
Right. And the peripheral blood.
Okay, I want to pause here. Let's take a second to do a quick recap and consolidate this for the listener because the implications here are massive.
Good idea.
So bullet point 1. Innate immune cells have been an untrackable blind spot because they lack nuclear barcodes like T cells have.
Right.
Bullet point 2, researchers finally cracked the code using the Mitotrek algorithm to filter the noise of fast mutating mitochondrial DNA.
The family recipe typos.
Exactly. And bullet point 3, by combining this with ATAC sequencing, they mapped not just the family trees, but the behavioral states of over 200,000 cells across the blood, healthy tissue, and 2 contrasting types of cancer.
Which is an unprecedented level of resolution.
So, if Mitotrek gives us the barcode scanner, what actually happened when they scanned the tumor microenvironment? Did these innate foot soldiers behave the way our adaptive T cells do?
They did not. Not at all. So the researchers looked at the clonal fractions, which is basically a metric of how much a single-cell lineage multiplies and expands its ranks.
How big the family gets.
Exactly. And they found a massive biological contrast. The adaptive immune cells, the T cells, B cells, and plasma cells, form massive clonal expansions right inside the tumor tissue.
So you see huge families of identical T cells locally multiplying.
Yes. They draw resources, they establish a massive local footprint right there on the battlefield. But the myeloid cells, the innate monocytes and macrophages, exhibited incredibly small clone fractions.
Meaning they don't multiply en masse once they breach the castle walls.
They simply do not. The data clearly shows that instead of local expansion, myeloid-dominated clones were the most frequently detected lineages spanning across all 3 sampling sites.
The blood, the normal tissue, and the tumor.
Yes. The innate immune presence in a tumor relies heavily on a continuous real-time supply flowing from the bone marrow and the peripheral blood.
Oh, wow. So if T cells are a local militia that sets up camp, recruits, and multiplies right on the battlefield, then our myeloid cells are more like a steady, relentless stream of individual specialized contractors constantly arriving from the highway of our circulatory system.
That is a perfect analogy.
Right.
And it completely flips the script on how we view tumor infiltration. Your body's perpetually shipping new troops to the front line.
But that creates a secondary mystery, doesn't it?
It does.
If these myeloid cells are constantly streaming into the tissue from the blood, what exactly is their operational role when they actually arrive? What do they become?
Well, to answer that, the researchers zeroed in on a critical subset of cells called the DC3.
The type 3 dendritic cell.
Yes. Dendritic cells are essentially the intelligence officers of the immune system.
They capture a piece of the tumor, process it, and present it to the T cells, right?
Exactly. They provide the exact coordinates for the strike. And DC3s are highly enriched in lung tumors and are extraordinarily efficient at priming those T cells.
But where do they come from?
Well, that's the thing. The developmental pathway of these specific DC3s has long been debated. We just didn't know which circulating cell in the blood acted as their direct precursor.
Until Mitotrek?
Until Mitotrek. Using that mitochondrial lineage tracing, they finally found the definitive link. Intratumoral DC3s are directly clonally related to circulating CD14 plus monocytes in the peripheral blood.
CD14 positive monocytes, meaning the generic monocyte floating in your vein right now as you listen to this is the direct parent of the highly specialized DC3 intelligence officer inside the tumor.
Yes, and they proved this using the second half of that complex sequencing technique we talked about, the ATAC-SAC, the chromatin accessibility mapping.
Okay, let's slow down and explain chromatin for a second.
Sure. So to appreciate how they proved this, we have to look at epigenetic memory.
Right.
When a cell differentiates from a generic monocyte into a specialized DC3, it doesn't change its underlying DNA sequence.
The genetic code stays exactly the same.
Right. Instead, it changes which parts of its genome are physically accessible. It unspools certain sections of DNA around proteins called histones, leaving them open for transcription.
While tightly winding up and silencing other sections it doesn't need.
Exactly.
Right, so if you were listening to this, imagine DNA as a massive library of blueprints. Epigenetics is basically which blueprint scrolls are rolled open on the desk and which ones are locked away in the filing cabinet.
That's exactly it. And what the researchers found is that the bookmarks placed on the monocyte's genome while it was still in the bloodstream. They remain glued in place even after the cell transforms into a DC3 inside the tumor.
The DC3s retain a distinct epigenetic memory of their monocyte precursors.
Yes. The physical 3D architecture of their chromatin still carries the shadow of their origin in the blood.
And that shadow is what allows us to track their functional evolution.
Which brings us to the most disruptive finding of the entire Liu et al. Paper.
Here's where it gets really interesting, folks, because we've established that the troops are flowing in from the blood and that monocytes transform into these crucial intelligence-gathering DC3s.
Yep.
But the ATAC sequencing data completely destroys a decades-old dogma in immunology.
It really shatters it, because the classical model in oncology has always been the blank slate theory.
The blank slate. Explain that.
The assumption was that a naive, completely neutral monocyte enters the tumor from the blood. It's a blank slate. And once it crosses into the tumor microenvironment, the local toxic chemistry of the tumor dictates its fate.
Like the local weather changes the cell.
Exactly. Depending on the local signals, it either becomes a helpful dendritic cell or the tumor corrupts it into an immunosuppressive macrophage that actively aids the cancer's growth.
So we believe the tumor was the ultimate local puppet master.
We did. But when the researchers analyzed the clones spanning the blood and the tumor, they discovered fate-biased clones.
Fate-biased, meaning they weren't blank slates at all.
Not at all. The monocytes still circulating in the peripheral blood, miles away from the tumor, already demonstrated strict, pre-programmed preferences for becoming either pro-inflammatory dendritic cells or harmful suppressive macrophages.
I want to use another analogy here.
Go for it.
It's like students choosing their college major before they even enroll, right? Rather than deciding based on what classes they take once they get on campus.
Yes. That's brilliant. They already picked their major on the highway.
But how? How could the researchers tell what major they picked?
They looked at the transcription factor motifs, basically the specific genetic control switches that were accessible in the open chromatin of these blood monocytes.
The open blueprints on the desk.
Right. And they found that DC-biased monocytes, the ones destined to become your tumor-fighting intelligence officers, already had specific pro-inflammatory genetic programs unlocked.
What kind of programs?
They were heavily enriched for motifs like NFKB, IRF, STAT2, and BLOMP1.
Okay, let's ground that in the biology because those are heavy acronyms.
Very heavy.
NFKB and STAT2 aren't just random letters. They are the master regulators of the immune response.
They are the alarm bells.
Right. Like for NFKB, think of the F for fire. It's pro-inflammatory. When those motifs are open and active, the cell is physically preparing to manufacture pro-inflammatory cytokines like TNF-alpha.
Exactly.
It is priming its surface receptors to sound the alarm. It is preparing for war while still riding down the highway. What about BLIMP1?
BLIMP1 is crucial for differentiation. It's basically the switch that commits the cell to a specific functional state. So having it open means the cell is locked and loaded to become a mature dendritic cell.
Wow. But what about the other side? The bad guys?
Conversely, the macrophage-biased monocytes, the ones destined to betray the immune system and help the tumor hide, were enriched for entirely different master regulators.
What are the blueprints?
Their open chromatin featured motifs for the MAF family, NRF2, and RUNX12.
Okay, and biologically, transcription factors like MF and NRF2 do the exact opposite of NFKB.
They really do.
And mnemonic here for you listening. NRF2. Think no response factor.
Oh, I like that. No response.
Right, because they transcribe instructions that produce things like arginase or anti-inflammatory cytokines. They physically create the suppressive blankets that calm down the immune response, exhaust the T cells, and allow the tumor to grow unchecked.
And RUNX12 works alongside them to lock the cell into that suppressive, healing-like state, which the tumor exploits.
And the crucial takeaway here, the mind-blowing part, is that these suppressive blankets were being manufactured before the cell ever touched the tumor?
Exactly. Which this raises an important question, perhaps the most critical question of this entire deep dive.
Which is how?
Exactly. If the tumor isn't doing the brainwashing locally, how are these monocytes getting fate-biased in the peripheral blood?
Right, they're miles away from the castle.
The biological implication is that the tumor must be releasing systemic signals, likely a cocktail of specific cytokines, out into the broader circulatory system.
So the localized tumor is actively projecting its influence outward.
Yes. Epigenetically preconditioning the bone marrow and the blood supply to ensure that the troops arriving at the gates are already compromised.
It is actively poisoning the supply lines.
This is a systemic war.
Okay, let's do a quick structural recap here to cement these massive shifts in understanding. We're at the final act here.
Let's review.
Bullet point 1. The Mitotrek algorithm proves that innate immune cells do not multiply locally like T cells. They are a continuous, mobile supply from the bloodstream.
Streaming from the highway.
Right. Bullet point 2. Crucial intelligence gathering cells, the DC3s, originate directly from circulating CD14 plus monocytes, retaining an epigenetic memory of their origin.
The bookmarks stay in place.
Exactly. And bullet point 3. The blank slate theory is dead. Monocytes are epigenetically fate-biased in the blood program to be helpful or harmful by systemic tumor signals before they ever reach the battlefield.
Which, this forces a radical reevaluation of how we design cancer therapeutics.
It has to, right?
Absolutely. For years, the pharmaceutical industry has poured billions into developing drugs designed to penetrate the tumor microenvironment and reverse the suppression of macrophages locally.
We try to wake up the corrupted troops inside the castle walls.
Right. But if their fate is largely sealed by epigenetic programming in the bloodstream, local interventions are arriving way too late in the biological timeline.
You're trying to convince a soldier to switch sides after they've already fired their weapon.
Exactly.
So what does this all mean for the future of medicine? We started this journey looking at a microscopic typo, a random mutation in ancient bacterial DNA living inside our cells.
The mitochondrial barcode.
Right. And by tracking that typo, we uncovered a systemic war.
And if the epigenetic state of a monocyte in the blood is being actively altered by systemic tumor signals, it provides a window into an entirely new paradigm.
Think about the diagnostic potential here.
It's massive. If these fate-biased monocytes appear in the peripheral blood, showing open chromatin for NRF2 or RUNX12, their epigenetic state could serve as a highly sensitive biomarker.
That is a staggering thought to end on. Because if the tumor is broadcasting its presence by reprogramming the blood, we wouldn't even need to wait for a physical mass to show up on an MRI, would we?
Not necessarily.
We could potentially use a liquid biopsy, just a simple blood draw, to sequence the chromatin accessibility of your circulating monocytes.
Just check the blueprints on the desk.
Exactly. We could detect the epigenetic footprint of a tumor's systemic signaling before the cancer is even visibly detectable on a scan.
It transforms our innate immune cells from a biological blind spot into an ultra-early warning system.
And maybe the future of immunotherapy isn't just treating the tumor. Maybe it's treating the blood itself, ensuring we're sending the right troops into battle from the very beginning.
That's the real frontier.
So next time you feel your own pulse, consider the millions of fate-biased cells currently navigating your circulatory highway, carrying the epigenetic blueprints of your systemic health. Something to think about. Thank you for joining us on this exploration of the Liu et al. Paper, and we'll see you on the next deep dive.